Good afternoon. My name is Claire Hope Cummins, and I'm going to be the moderator for the panel we're starting right now. I've been covering genetically engineered organisms, GMOs as we know them, for over 20 years. And during that time, many of the people in this room, those of us here on the stage as well, have worked very hard to try to stop them, to slow them down, and to label them with varying degrees of success. And during that time as well, the proponents of these transgenic technologies have been working really hard in their labs. And as you well know, they've been very aggressively promoting their own agenda for what we're calling false solutions. We're going to be talking about more false solutions that have come about, what we call new and emerging technologies. This is beyond GMOs. And we're going to tell you about them. We're going to give you some news. Ordinarily on conferences like this, you don't get news, but this is really important information that we're going to relay to you about what's going on, what's coming up. And it's not just about these new technologies, about what you need to know and why they're dangerous or what's going on with that. It's also about our movement. It's about what we do with this information that you're going to get today, and how we work together, and whether or not we use this new opportunity that's coming up to work together more effectively and to put into place our own vision for ecological social justice. So here's how the panel's going to work. We've divided roughly into 3 sections, about 20 minutes each. And we're going to start with an introduction to the new technologies. We're going to tell you what they are and a little bit about how they work. And then we're going to go deeper in section 2 to talk about the issues and the challenges and the problems and the kinds of things you really need to know in your work about what you're going to be seeing in the news about them and how they pose problems in many areas, such as agriculture, the environment, and social problems as well. And then the 3rd section we're going to talk about, again, we're going to come back to our own movement and how we respond to this, what we're going to do, and how we're going to implement our vision more effectively and use tools like real solutions like agroecology and what we're going to do moving forward. I happen to think that these new technologies, instead of actually just being alarming and another problem we have to deal with, offer us a real opportunity for us to take this on in a new way and to move forward together, being more effective. Some of you in the room know that I've actually tried to retire. I know, the gray hairs, but when I found out about gene drives in particular, I was so alarmed. I got very fired up, and I came back in, and I'm up for this. And I hope that you're going to feel the same way. I think we have such a passionate and expert panel for you today that you're going to leave this workshop also totally fired up about what can be done. I'm particularly concerned about gene drives and how they might be used in Hawaii. And I think we have some people from Hawaii here that'll also be, where are you? Hello? Yay, good. And then we're going to have a workshop. The speakers have agreed to be available right after this session outside if you want to talk to them individually. But we're going to have a workshop at 5 where Dana and I will be able to go into much deeper both the issues around the technology and our response to that. So let me introduce to you our panelists. Right here we have Gopal Dayaneni, who is with Movement Generation. Yay! And Monica Moore, who's the Program Director for New and Emerging Technologies at the CS Fund. And Dana Perls, who's a campaign organizer for Friends of the Earth US. And Maywa Montenegro, who is a great scientist who's going to really hold up the science end of the panel here. She's a fellow at the Berkeley Food Institute as well as at the National Science Foundation and a PhD candidate at UC Berkeley. So let's just give them all a hand. Absolutely. We're going to start with a brief introduction to new technologies, and to start us off with that is Dana Perls.
Hi, thank you, everyone. So I just want to start out really emphasizing we're going to be specifically talking about this next engineering. You might hear it called GMOs 2.0 or synthetic biology, but this is part of a much larger context of emerging technologies, new technologies that are racing forward at such a rampant pace that we're really struggling to keep up with what is actually happening. And just to say that there are a lot of risks and a lot of concerns that we need to know about, regardless of whether or not we are PhD scientists or the regulators themselves, that it is our duty that we need to stay on top of this. We need to be recognizing the concerns, and these are emerging technologies that affect all of us, and therefore we need to be the deciders of what moves forward and how and what is the world that we want to live in. And so specifically, we're going to be talking about a new bucket of technologies referred to as synthetic biology or gene editing, but it's the next generation of genetic engineering. Genetic engineering has shifted, and now we have to shift with it, right? And it raises concerns that go far and beyond GMOs as we know it. So one of the things that we need to know are that this is different, it's much more extreme, it's a tighter level of intervention at our very levels of DNA, right? So now we are looking at a consolidation of corporations looking to redesign DNA and profit from this redesign of DNA. Already it's expected to be a $40 billion industry by 2020. And just to put it in context, this next generation of genetic engineering is not just confined to agriculture and plants. We're going to talk about that mostly here in terms of how it relates to conservation and agriculture. However, in the bigger context, this is being used as a platform to look at creation of bioweapons, agriculture, redesigning plants, insects, animals, all the way to applications to humans, GMO humans. So to note that this is a much larger context, and we're going to focus quite narrowly here today. So I just want to say really quickly, how does it work? GMOs, as we've known it, has been more about inserting the DNA of one existing organism into the DNA of another existing organism. And this is much different. This is about designing DNA that doesn't exist anywhere, brand new DNA. This is about reprogramming organisms, new life forms, getting organisms to do things that they wouldn't normally do by changing the way that they have their instructions, their natural instructions. And that can involve, as I said, creating new DNA, silencing new DNA, deleting DNA. And I'll give you two examples really quickly. One common example that is being used to create new plant substances in our food and cosmetics and cleaning products is that you would ultimately sit down at a computer, you can type out a new DNA sentence, print it out with a DNA processor, insert it into an organism like yeast or algae, and then through a process of fermentation, you can produce things like synthetic biology vanillin or stevia or ginseng or coconut oil or breast milk or whatever it is that you have designed this to do. That's one example. The second example is one that Claire mentioned, which is the gene drive. And this is actually an extremely scary technique. This is about designing an organism that will spread into an entire species. I'm just going to give you an example. Imagine releasing a single fly, one fly, into the wild, and that you could, by doing so, genetically alter all flies on the entire planet. You can get them to turn yellow, you could get them to release a toxin, you could drive them into extinction. So this is a new and very controversial type of genetic engineering technology that can permanently alter an entire species by releasing one bioengineered organism into the population. There's this assumption that this power could be proved useful. However, this is a power that may cross a moral and ethical threshold that we need to know about, and we also need to discuss and decide whether or not that threshold should be crossed at all. And the applications of this gene drive that we'll be talking more about, we're seeing proposed in many uses ranging from bioweapons to daughterless mice, so that we could drive mice out of existence, particularly on islands, or for example, re-engineering weeds so that they're only susceptible to the pesticide that you've engineered them to be be resistant to or susceptible to. So this really is a drive to further control nature and patent it for a larger profit. So that's kind of a very broad brush of what this is. It's a larger, deeper intervention at our cellular level, and it's this attempt to engineer nature and to regulate and to control nature with an engineering principle as opposed to a biological principle in our life. So I'll pause there.
So we're gonna go to each panelist and ask them to address how they came to the work on new and emerging technologies and their own perspective on the introductory remarks that Dana made. Maywa, would you please come speak to us?
Sure, thanks for that, Dana. So I came to genetic engineering, I guess, actually as a biologist in a laboratory. As an undergraduate, I worked on agrobacterium, which is the microbe that's often used to transform plants to integrate new genes. And I found myself really enamored with the idea of using this as an application for potentially feeding the world, so to speak. So I thought, you know, if we could biofortify cassava or banana, that would be a beautiful strategy for helping alleviate malnutrition and hunger in the developing countries. So for several years, I thought I was going along that pathway to be a PhD in plant molecular biology. But I took a detour, and I spent several years as a journalist interviewing many folks who come at the problems of quote-unquote feeding the world from many diverse perspectives, and I encountered people who work in agroecology and in food sovereignty, and at around 2011 that kicked me back to graduate school, where I've been learning a lot more about the history and politics of the food system. And now I spend a lot of my time in social theory, but also trying to connect more to grassroots movements, farmer organizations, indigenous communities, and to actually reconnect with my original roots, which are, you know, based in Peru. I'm half indigenous Peruvian and half Dutch, so I sometimes joke with people that I'm a hybrid seed, half colonized, half colonizer. But that's kind of where I come to this position from.
So do you want to tell us from your perspective about the new technologies and introductory perspective?
Well, I believe that one of the difficult things to wrap your mind around when it comes to a genomic editing tool like CRISPR is that it's not a single thing. Agrobacterium was a pretty blunt instrument. We took this bacterium, and it has a natural circular piece of plasmid called the TI plasmid. And it uses this, if you've ever seen trees outside with this crown gall, that's agrobacterium, and it creates cancers in trees to feed itself. And geneticists used to harness that to inject foreign DNA. And it was pretty random, so they had to try many times over. With CRISPR, we have the ability to design strands of RNA that, like a homing device, can send it to particular places on the target genome, and it works in pretty much everything they've tried it in, which is spectacular if you're a molecular biologist, because you're pretty lucky if you get it to work in bacteria. And it works in plants and fungus and mice and people. And, but it can do an amazing variety of things, and I think oftentimes in the media we see it described as being something that does not introduce foreign DNA, and so I think it's important for people to realize that it can not introduce foreign DNA, but it is also very capable of the traditional, like old-fashioned, you know, gene splicing. It's very adept at that, but it can do also kind of microcosmic nips and tucks. So I think rather than throw our hands into the air and say it's something very precise and new and elegant in that it doesn't introduce foreign DNA and therefore we shouldn't regulate it as GMOs in the traditional sense, I think absolutely we have to recognize it as GMO, but not as a single GMO. And we need to kind of create regulations that are specific to the types of mutations involved, because they are not all identical. And so that requires a kind of deeper conversation with the public and with the researchers who are, of course, pretty aware of the different types of mutations involved. So I'll just start with that.
Gopal, I see you nodding your head. Do you want to add?
No, I guess I'm next.
Yes.
She asked me to come. Hi. Hello, everyone. This is Gopal. I don't know. I'm going to have to lean over, I think, or maybe I
Do you want this here?
Are you all getting a weird echo?
No, we are. I turned.
Oh, it's just us. Okay, great. I can live with it. Hi, my name is Gopal. I'm with Movement Generation Justice and Ecology Project here in the Bay Area. Yeah, how did I come to this work? Specifically looking at emerging technologies, and in particular synthetic biology, and now one of the things that we're going to talk a lot about is gene drives. So I actually came to this because I was working in environmental justice issues in the high-tech industry. I was looking at the relationship between technology and environmental human rights issues, including worker rights issues. I worked on the toxic waste dumping of electronic waste in prisons and using captive prison labor to do electronic waste recycling, quote unquote, green jobs. That was a joke. There's nothing green about using captive human labor to do anything. And so I really come to it from the perspective of actually looking at the justice implications and the relationships of power in technology. And that's really how I came to these issues. And as we started working on electronic waste and traditional technology issues, and then we started to see nanoscale engineering and nanotechnology emerging, and then because of all the big data, artificial intelligence really accelerating, and realizing there was this interesting neck-and-neck race to Armageddon between artificial intelligence and biotechnologies, and we're like, wow, what do we do? And so that's how we sort of came into it. And one of the things that for me has been really important is in this work and how we think about and assess these technologies and just orient ourselves to them is to not get stuck in the narrow view of a technology only being understood through its application. So we have a tendency, I'll use just a quick metaphor. We used to only talk about the internet and computer technologies in terms of the quote-unquote digital divide, in terms of the relationship, the user-end relationships to computers. Some people had greater access, some people had lesser access, and there was a justice issue embedded in that. And what was made invisible by that are all the embedded relationships of power that got the computer into your lap in the first place. And particularly here in the U.S., in the West, and particularly I think I can safely say with this audience, it is easy for us to, in an almost self-centered way, only think about application and to not actually think deeper and look deeper. And so that's how I came to it. That's what I'm interested in, and and there is nothing more to me, moving faster and consolidating relationships of power and creating technologies of control than what we are seeing with in particular, synthetic biology, now gene drives, and some of the enabling instruments of that, like CRISPR. So we'll come back to all that a little bit more with some detail, but yeah, I came to this work interested in power, and in particular, racial and economic justice issues.
Monica?
Oh, sorry.
I do need a timer. Hi, I'm Monica, and there is a funny echo up here, so Hello, 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 hello. I'm coming to this work informed by about almost 30 years work on pesticides, the problems of pesticides, and also genuine solutions to the situations that pesticides are supposed to impact. That was when I was working with the Pesticide Action Network, which really, and the fantastic people around the world that taught me so much in so many of those areas. For the last 6 years or so, I've been in a different but related path. I now work for a small foundation that has been funding in the area of a number of problematic technologies. They were one of the first funders of people's responses to biotechnologies back in the 80s. They funded PAN for a long time, still do. And they're now working a lot with people concerned about emerging technologies, and that's the program that I'm involved with. And so one of the things about looking at emerging tech for me, it's a bit deja vu all over again on one side. Many of the things that are coming up now seem very, very familiar in the sense of all the problems that are going to be solved, the excitement around it, the buzz and the hype, and some things are genuinely quite new, capacities that haven't been on the table, in the lab, in the marketplace before. So there's both a lot that's familiar and a lot that's changing very quickly. I was part of efforts to constrain and deal with emerging biotech in the 80s. And I think that a lot of what we learned through that struggle can be useful here. I also think that we need to train ourselves now to build on those strengths but see beyond them and work beyond them. A lot of people call, for example, synthetic biology geoengineering 2.0, and I think that's apt. We should remember, though, that emerging technologies covers a whole swath. I mean, it's a huge field. It's not just synthetic biology. It's not just any one thing. There was a time when some of the things that we take for granted now were emerging technologies. So we have to kind of prepare ourselves to deal with both these very specific threats, but also be able to assert more about what we need and what are genuine pathways and solutions to get there. So in addition to the specific things we're going to talk about with gene drives and synbio, I want to put out a couple of things. One is that the rate of technological change is speeding up, and that can be very overwhelming. And I want to just counsel all of us, myself included, on a daily basis to breathe deeply and remember that we can deal with this with what we know. As Gopal pointed out, if we've trained ourselves to see power relationships and be open to new approaches and alliances, we've got this. We can deal with this, and we can assert what we genuinely need and see through the smokescreen of what's being offered because someone controls it or could make money from it. Another thing is to recognize that a lot of technologies are converging. So, for example, synthetic biology is basically a merger of biology and engineering, full-on merger. And the idea is that the rules of engineering will run the game. That it's a game, life, a game, and that it's subject to the rules of engineering. So you design for optimal function. You know exactly what's going on. You have interchangeable parts you can scale. And actually, this is not how biology works. This is not how nature works. And one of the things that's very humbling to anybody who studies ecosystems or natural systems is how much we learn when things go wrong that we didn't know. Mm-hmm. You know, it's like a forensic exercise of, oh, who knew? You know, and things we used to call junk DNA, turns out they're not junk. So it's a precautionary note to start this out, both how we got into this and my approach to it is dealing with the specifics, remembering what we've learned along the way, and also that there's a whole lot of stuff that's coming at us pretty quick that we're going to have to deal with, but that we can, and to put in, this is what we also need is technology assessment that isn't just run by the marketplace. Currently, technologies are assessed by fundamentally laws of proprietary ownership and then whether they can make money and at what degree they can make money. And so there really needs to be, and I'm happy to be involved in a number of efforts to bring different parts of society together to say, what's our problem? A classic example, climate change, not going to be solved by the market, not going to be solved by the market. What do we really need and what's going to get us there versus what is going to make whatever interest or sector wealthy in the short or longer run, and second of all, what different groups with different interests say is feasible, either technically or politically, more importantly. And so that joint, inclusive, comprehensive technology assessment that includes the social and economic impact that isn't just run by the market is definitely what we need. It's an exercise you'll all recognize in power building, and at the foundation that I work for now, we're quite interested in helping bring that into the world. So I'll stop there for now. Back to
The time has sort of come to move to more specificity. This has been wonderful, good setup. And I would like to ask the panelists another question, which is sort of going further into the problems that you mentioned. What are the risks? What are the perils? What are the challenges both around the technologies, but also the challenges to our movement? And I maybe think we should talk more specifically about gene drives. I think that might give us a focus. Again, we'll have a workshop at 5, where we'll be there to discuss things with you, and the speakers will be available after the panel, because this is a little tricky stuff, and if you don't understand it, we're here to help make it plain. So, Gopal, we want to start with discussing the problems and challenges that we're facing with these new technologies. Here, you can have this one.
Yeah, I'm worried I'm gonna break that if I try and remove it. So I just won't touch that. Yeah, so I'm going to start with a broad philosophical assertion. You know, it's been named a number of times, but at the heart of the danger here is this idea that we can impose industrial order to living systems. But even deeper, the central violence of these technologies, in my mind, is actually in the mind frame, in the metaphor. It used to be that life was the metaphor for everything, and now machines are the metaphor for life. We talk about our minds as being hardwired. We talk about our bodies as engines. We talk about DNA as code. We talk about cells as chassis. And when you talk about DNA as junk, we talk about the disruption of living systems and the shackling of them to industrial platforms and engineering technologies as editing, right? We talk about deletion and insertion. And this isn't just a metaphor that makes it easy for us to understand. It changes the very way we understand. It erodes our ability to lean into the complexity of living systems, to look for complexity, to remember that the defining feature of living systems is relationship and the complexity of relationship. And so, from my perspective, it actually begins with the sort of cultural and cognitive assault that is taking place every time one of these technologies are put out and promoted, and the way they are promoted as we will cure malaria, they're all silver bullet solutions, right? And the silver bullet, of course, we're fond of saying, think twice before you jump in front of a silver bullet. You will get shot. And for us, we start there at Movement Generation. We want to actually confront and disrupt the machine metaphor and start to make meaning of the world around us in the ways that we always had for almost the entire history of our time on this planet, which is through living metaphors. So that's the first thing, the first problem with these technologies from my perspective. Then, so we talk about this in terms of, and I'm going to talk specifically about, so as an example, gene drives is a really good example. We are talking about population scale engineering, right? We're talking about the idea that you can actually control the trajectory of evolution. And this, the presumption that you can actually bring that much precision and that much order to the world is against our actual lived experience. We have to actually school ourselves out of recognizing that that's just not the way the world works. And in fact, we have lots and lots of examples of looking at ways that we've done that in the past that didn't work. You know, like the idea that we're going to deal with the I don't know, frog problem by bringing in rabbits, or the, you know, the various permutations of all the ways Australia went sideways through colonialism, and colonialism itself being a perfect example. The other thing is actually that the interesting thing about these technologies is that they don't have to actually work to be profitable. Right? Our friends at the ETC group make this point a lot, which is the promise that we will cure malaria, or the promise that we will be able to address conservation issues. One of the big false promises that we're really scared about is the way they're going after our interest in environment and conservation and biodiversity. That we can actually address invasive species by eradicating them from the population through gene drives, or this idea that we're going to be able to create algae that will synthetically engineered microbes that will do environmental remediation, or even that will sequester carbon. They all play into claiming to solve real problems that we're dealing with and that we're all concerned about. And so we need a way of actually looking at whether or not they can and won't do that. And it's time for me to wrap up, because I got a nice note that says time to wrap up, which I appreciate. The very last thing I'll say about that is that if you can see that a problem either creates a new environmental or social, technology creates a new environmental or social problem, or exacerbates an existing environmental or social problem, you have a pretty good idea that it's a false solution. So that's like a simple test you can do. Like, what's the trade-off here?
Maywa?
Thanks, Gopal. I wholeheartedly agree, and I am going to be a little bit less poetic, but drill down into maybe some of the technical problems that I see, but they are very closely linked to this idea of industrialization and the concept that we can apply the concepts of engineering and design to exert kind of human control over natural systems. I'm a journalist, as I told you, so a lot of learning that I do is scrolling through newspapers and magazines and seeing how is gene drive, or how is CRISPR and genomic editing depicted in the media, because the media often serves as this interface between researchers and the general public. So how we are kind of interpreting the stories that we're telling each other is often filtered through what journalists write. And I just came across one from late 2015 that resonates very closely with what Gopal was saying. Gene drives give scientists power to hijack evolution. And this is a headline as if it's a good thing, and it's a beautiful thing to give scientists the power to hijack evolution. And I'm fascinated by that being billed as a benefit. So just briefly, a gene drive, which is the kind of one that we'd like to drill down into a tiny bit, is nothing new. These occur in nature, and researchers have known for a long time that in general, when you have two plants or two animals that mate, you'll get roughly 50% chance of inheritance. But they looked around and they said, wow, something is happening in nature that this gene is appearing much more frequently in these populations. And then sometime in 2003, Austin Burt at Imperial College in London said, I think that there is probably a way that we can use science and the tools of molecular biology to make this what they call jealous propagation, like using Richard Dawkins' term, the selfish gene, jealous propagation of the genes into the next generation. I think we might be able to make it happen in the lab. And he wrote a paper, and it was very widely taken up in 2003, but it wasn't really possible for about a decade until CRISPR came along. And basically what happens is, instead of genetically engineering single organisms, a gene drive sweeps an entire edited population exponentially because one chromosome can copy itself onto the partner chromosome. So you get both, and that's called a homozygous condition. So anyway, now there are research labs at Harvard very interested in how we might use this, for example, to combat diseases like West Nile and yellow fever and Lyme disease. Zika has come up as a potential use of gene drive. And then ecologists and many science fiction movie watchers say, worst idea ever. So I have some notes here. This is not my own language, so I want to attribute it to, he's actually the editor of a rheumatology journal who wrote a beautiful little essay on CRISPR, and he said, altering an entire population of mosquitoes or eliminating them altogether could have drastic and unknown consequences for an ecosystem. When you remove one pest, it might make space for others to emerge. It could affect predators higher up that used to eat that for their food, so you might wipe out that organism, and that could cascade. Hypothetically, that guide RNA that I mentioned previously, that can mutate too over time. So you're then targeting different parts of the genome. So that other mutation that you didn't intend could race through the population in the wild. And I'm going to wrap up soon. The other big question people have wondered about since CRISPR is so much faster, cheaper, and easier to learn is the potential threat of bioterrorism. And this is not exactly my greatest fear. I think we can fear a lot more displacement of traditional varieties than bioterrorism, but it is a potential if it gets into the wrong hands. My greater fear is kind of the hubris that is involved, that we would say, as a matter of first order, that we have the authority to doom a species to extinction just because it is something that is consuming our commodity crops. So I'll end with that one.
Dana, Dana, is this on?
No.
Now it is.
Thanks. Nope.
I think so.
Dana, sorry, before you start, I wanted to make one follow-up on, and we can do a little bit more follow-up in this session. We're supposed to, we thought we might, on what you've just said, Maywa, and that is, from my perspective as a feminist, they have proposed a gene drive which creates a daughterless mouse. And basically what would happen, it would drive that trait through the generation so that everything would come out male. There would be no daughter, so it would be a daughterless mouse, therefore wiping out that species of mouse very quickly. And it seemed to me that a daughterless anything is such a weird idea. I mean, Gopal was talking about metaphors, and we've been talking about language. Just think about that. If that's okay with you, that we as one species have the right to eliminate a whole species by wiping out the female and overtaking sexual reproduction in that way. So that's one point, just to illustrate what's been said. The other point, as a lawyer, I'm really concerned that we never, in the 30 years we've been dealing with transgenic technologies, have ever taken a good regulatory look at this. And the people who are trying to propose gene drives in conservation are saying we have a mature regulatory system here in the U.S. and everything's going to be okay. We have no regulatory system in the U.S. or anywhere else for that matter. So it's a big vulnerability that I wanted to add into our discussion of all the problems and perils around this technology. Dana, do you want to follow up?
Sure, yeah. So I'm going to be really specific also to agriculture, since we've heard some of the conservation tools. The first thing that I want to point out that we didn't necessarily I didn't make clear, but so that people understand that this is a technology platform that you don't need to go and start a campaign on specifically. It is going to come to whatever issue you're working on, and it already is. Whether you work on soil science, on food, whether you work on animal welfare, whether you work on oceans, this is a technology platform that is coming to try and propose a techno fix for what you work on. And it's really critical that you understand then the risks of what could be involved. And then the other thing just to note is that we are unleashing this set of technologies with very few ideas about what the consequences are. We have ideas of what the consequences are, but not to have too many double negatives, but there are a lot of unknown unknowns that we've heard said on this panel. So in the context of agriculture, I would name that there's concerns around contamination, around perpetuating the industrial agriculture systems that bring us further away from agroecology, and that we are also risking the livelihoods of hundreds of thousands, if not millions, of small farmers around the world, and I'll explain why. So one of the techniques that I described before, where you type in a genetic code into a computer and you re-engineer an algae to produce stevia, vanillin, ginseng, this is an attempt of the industries to bring food from laboratories to our tables that would replace the products that farmers across the world produce in sustainable ways. That you could order vanilla from a lab rather than from the rainforests of Madagascar. That you could get your coconut oil again from being squeezed out of an algae in a vat rather than going into the sustainable farms in Indonesia. And so there's an impact on livelihoods. I also want to say that the perpetuation of large-scale industrial agriculture is at risk here, because you have to feed the algae and the yeast something, and you feed them sugar, you feed them GMO corn, you feed them sugar cane, you feed them sugar beets. So we're now talking about switching over from land for food to land to produce feedstock to feed an algae to then produce the food. So this is again a corporate control of the entire system of agriculture. And then lastly, contamination. When you genetically engineer algae, for example, which is one of the most prolific organisms on the planet, to produce, let's say, petroleum or biofuel, and that gets out into a waterway, there's not going to be recall. You can't recall organisms that are designed and specifically meant to self-replicate. Likewise, with the gene drive, if you have just engineered a seed so that it will only be susceptible to Roundup, or a seed that will go that will drive a weed out of existence, there's no recalling that afterwards. We have permanently altered our ecosystems, and in a way that we don't know how is going to affect the rest of our environment. And then lastly, I just want to say the other risk that's really critical amongst many is corporate control. Already we're seeing a multi-billion-dollar company named Intrexon. They own the GMO apple, they own the GMO salmon, they own the GMO mosquito, they own DNA cloning companies that for cloning animals. They own a DNA fabrication company so that they can then be the one that sells everybody the DNA to make new life. Right, so we are seeing massive consolidation of companies to figure out how to control and profit from the redesign of DNA. And so just imagining the very specific risks that go along with that, and the risks to the visions that we're all gathered here to discuss, which is something more natural and agroecological and sustainable, not just sustainable for our environment, but for the lives of people on this planet.
Something that Dana just said reminded me of a blast from the past. Many of you may remember that when biotechnology first came along and transgenic plants, that the herbicide tolerance was the big thing, and the idea behind getting Roundup Ready everything was because literally Roundup was considered a safe herbicide with no environmental or health impacts. And so it was so much better than the old stuff. And now we look at that argument, and now that there's Roundup resistance because of the widespread use, they're talking about bringing back some of those old, more, what were back then considered more dangerous because the Roundup doesn't work anymore. And so you sort of see how the cycle doesn't get broken. That's a digression. Let's see. I want to talk about on the gene drives, drilling down a little bit more on what my friends and colleagues here have said, which is the myth of precision. And there's a wonderful essay on this that we can make available afterwards. But basically the idea that you can target something so tiny, incredibly precise, means, targeting something precisely is the same thing as understanding what the consequences of that will be. They're not the same thing at all. It's like understanding at nanoscale. It's very, very, very small, but the implications can be huge. And the ability to target is not the same as the ability to understand. And I would just caution everyone to look for that when people are telling you, or when you're hearing how very, very precise this is. The implication is, and therefore the consequences are well understood because it's so precise. It's like one does not equal the other. Another thing, I do get mad. We were supposed to say, what keeps us up at night. I don't want to go into those dark places, but I get mad when I see these same bankrupt arguments that have proved so damaging to so many people and to so many ecosystems around the world coming back around again with even more powerful drivers, which is not to say that nothing good can ever come of any of this. It's not. But haven't we seen this movie before? I'll give you an example of sort of the precision idea again. Some people are saying in the genetic conservation of seeds, they're saying, it's so expensive to run seed banks. If we can just identify the code, the DNA code that characterizes different seeds, then all we have to do is store that digitally, and we don't have to have this huge infrastructure around saving seeds. Does that not sound like a just fundamentally terrible idea that's antithetical to farmers' interests and people who rely on farmers around the world? So these things start getting presented because of the precision, because we can understand and map out these things in a digital content. It's not the same as understanding consequences. Let's see. One other thing I would say, time? Okay. Dana mentioned that feeding the perpetuation of industrial scale, very destructive agriculture doesn't go away if you go this route because you'll need to feed the microbes sugar or whatever. Another fuel source has been proposed, which also speaks to the convergence of a lot of these technologies, which is methane. Turns out you can synthetically alter organisms so that they can be fueled by methane. There are living creatures on this planet that actually use and live on methane. And so there's been some real interest exploring how to use some of that methane that comes out of the fracking industry for this. So I'm just saying that the traditional limits or barriers between both solutions, genuine and false, break down under this model. And so it means that we have to be a lot more careful. And if you suddenly start hearing the fracking proponents say, hey, this is a great fuel source for a less carbon intense footprint because we're helping feed people, antenna should go up. Okay, I will stop there.
So we just have a few more minutes. It's so rich. Thank you all. But could we then just maybe go down the line and give your vision for a positive sense of where our values can be brought forward and the work we do as a movement, looking at these technologies, facing these challenges, the false solutions, and bring up your attention to the real solutions that we can implement effectively as a movement. Let's see, we're going to start with Maywa. Yeah, Maywa, would you start, please?
Sure. So I think Dana said earlier, like it or not, CRISPR is coming. It's already here. We not only have daughterless mice, but there are corporations working on Brazilian beef cattle for larger muscles, chicks that lay only female offspring for more egg production, beef cattle that have only males for more efficient meat to bone conversion ratios. So those are in the product pipeline. What I'd like to do is kind of pivot a little bit and say that what I think, since CRISPR is here, What the food and farm movement could really do is seek to redefine the dominant criteria for evaluating all sorts of crop improvements. I put improvements in quotes, and this includes not only CRISPR and the next generation, but all forms, including conventional breeding. So some of my colleagues and I have begun to brainstorm some criteria for social and ecological acceptance of GE, or genetic engineering. I won't go into them in detail, and maybe at the workshop I can describe them more. But for example, does this technology achieve or satisfy distributive justice standards? Are the cost, benefits, and risks of crop improvement shared equally among populations, so farmers and consumers, across geographic scales, including developing countries and highly industrialized countries? As we know, the Green Revolution posited that it would be helping to feed the world, but it actually mostly benefited the larger, more capitalized farmers. So if we put a technology to that test, does it meet distributive justice? That it should be developed, introduced, governed in transparent, participatory, and publicly accountable ways. And very importantly, that users of a technology should be able to withdraw their consent. As we all know, most farmers and consumers now are often locked into use, and consumers cannot even see by looking at a label whether they're consuming a genetically modified organism or not. That the technology should contribute positively to the conservation of agrobiodiversity, and I mean land races, wild relatives, heirlooms, other sources of genetic diversity, and a corollary to that criterion, of course, is the cultural knowledge, the linguistic knowledge, and the other forms of epistemic or knowledge diversity that go into the conservation of genetics on the land. And then the last couple, the technology should be free from intellectual property. We should avoid structural lock-ins, and that the technology is consistent with food sovereignty.
Nailed it.
I'd like to just say ditto.
Yeah.
That pretty much sums it all. I think that, you know, just to emphasize transparency and a truly democratic approach is not what we're doing. I don't want to purchase food, for example, that is labeled non-GMO when actually it is using these synthetic biology techniques. I don't want somebody to say, this is going to be beneficial. I want to say, well, is this beneficial to who? This is beneficial to the company that is controlling the technology, but is this beneficial to the farmer? And so in general, I have a vision of great transparency, and that's going to bump heads with confidential business information and proprietary rights. But I have a vision also of the agro-ecological systems that Maywa and others earlier today have been talking about. What does that truly mean to be sustainable? Who are the people involved? What is the cultural knowledge involved? What does it mean for our entire environment? And how are we looking at things holistically? It seems pretty simple just to say, let's look at things holistically, but it's not how we currently do things. You really did say all of the visions that I would have. So I'm not going to repeat them, and I'll just move on to Monica.
I'm actually really excited by the progress that many social movements are having, advancing genuine solutions, and I think agroecology comes to mind. Not that there aren't tremendous obstacles, but the wonderful thing about genuine solutions is that they really work, actually. And at a time when that's what we desperately need. So in the area of technology assessment, I'm also very excited by the coming together of different social movements around the world. It's a positive example of global exchange of ideas and innovation, from climate justice, from agroecology, from women's rights, from movements of landless people, indigenous peoples, and looking at the need to get ahead and not just technology ambulance chase, but to say, okay, what's on the horizon? What's coming at us? What do we think of it? What do we need? Is that really going to do it? And then have somewhere to go with that. Those conversations actually are going on and have been going on for, I'm going to say a decade. And they're now coming to life. The United Nations has recently said that they've green-lighted something called the Technology Facilitation Mechanism, which gives a political platform and a way to reach governments around the world that they have to listen, where very, very skilled social activists and movement leaders can bring these concerns forward and give them a stage that they haven't had before. So it's very exciting work. I'm also going to say I'm excited by the folks dealing with for example, the organic space, preserving that, and the non-GMO people are taking this new threat very, very seriously and talking together, and I'm encouraged by that positive work. So there's lots of really deep, powerful building work that's going on in addition to all these scary things that we learn about on a daily basis. I'm going to slightly out two of my colleagues and say, I don't know if they were going to mention it, but Dana and Gopal have fantastic handouts. Probably not enough for everybody, but just going to say, really good stuff, people. Go for it.
So I get to go last on this one. So I want to start by an axiom of systems that I think is useful to remember as we talk about the vision of the world we want and how we're going to get there. The first is that the purpose of a system is only determined by what it actually does. The purpose of a system is never determined by what it aspires to do or claims to do or wants to do. That you actually can only understand a system's function by observing what it actually does, and that is in fact its purpose. And without doing a whole political education on my wonderful handout, quote unquote, I want to say that the purpose of the economy that we're in is the concentration and enclosure of wealth and power. And all wealth is generated through the work of the living world. All wealth is generated through the work of the living world. From the bacteria in the soil to the black lives that made this economy what it is today, from the insects that pollinate to the indigenous people that hold the knowledge that we need to transition to the world that we want and to remember our way forward. That is where all wealth comes from, and the interesting thing about when we talk about biotechnology and we talk about the corporate control of agriculture, we often talk about the erosion of biological and cultural diversity, the loss, the assault on seed, soil, and story as a consequence of this corporate concentration. And it is not. It is a strategy of corporate concentration. It is the eradication of diversity that makes it easier to control and concentrate power and resources. And so when you only have a few varieties, it's much easier to control them all. And so in that context, so I'm a big fan of the technology assessment conversation. I think that's a huge part of what we need. But the way we assess it has to look deeper than just the consequences of deployment. And so we talk about a set of criteria for what constitutes a real solution versus a false solution. And the assessment that we're trying to make is alliterative, because that's what we do really well. And it's the idea that we should be looking at root cause remedies, rights, reparations, and real representation. And so root cause remedies means if somebody says we're going to deploy a gene drive to eradicate mosquitoes in Africa, and not surprisingly, all of the sympathetic deployments are about island nations, indigenous peoples, and Africa, where they want to test these things. So instead of saying we're going to eliminate malaria by eradicating mosquitoes with a gene drive, we need to step back and ask ourselves, why is malaria a problem? What are the roots that drive the inequity, the inequitable distribution of suffering on this planet, and how do we start to address those? What are the economic foundations, the social, colonial, political, cultural foundations that create these problems in the first place, and how do we address those? So root cause remedies. Second, rights. Collective rights, human rights, rights of Mother Earth, the rights of indigenous peoples. rights should be the basis of how we understand and assess the value of tools and technologies in addressing them. If they violate those rights, then there's a problem there. And there is a struggle in our culture between rights and regulation. And I won't get into it, but like the whole thing of like the regulatory system is about determining how clean your water can be. And rights of Mother Earth and rights of nature and our rights is the right to clean water. And those are not the same thing. And so we must base our assessments and our movements on the assertion of rights. And then reparations, that is just not recognizing past harm, but repairing the nature of our relationships. So moving forward, we are living in right relationship with each other. So that's an important thing. Does a technology concentrate control, or does it change our relationships forward going? And last but not least, people should have control over the decisions that affect their daily lives. You should not be allowed to deploy these technologies on communities without them having control over the decision to do it.
Let's thank our panel, and thank you very much for attending. Thank you. Thanks for the panel. Come on, big hand for the panel. We're done.
And just to plug that we're going to do a deeper dive conversation at 5 o'clock in the Emerald Room. And if you, after the next panel is done, everybody's going to be available outside to talk immediately after in case you can't make the talk at 5 for the workshop.
Thank you for saying. Thanks for hanging with us. Thanks for hanging with us for so long.